Good afternoon, everyone. Welcome to the ABRF webinar series. Today, we're delighted to have our colleagues with Curio Biosciences. And before we turn it over to them, I'm just going to give a couple of housekeeping reminders and share some information from ABRF. Just a second. All right. First of all, feel free to introduce yourself in chat. We'll be using chat for questions. So if you have questions for the panelists, please put them there. They'll be addressing those near the end. But again, if you'd like to introduce yourself, where you're joining us from, your institution, your location, that'd be great just to see the range of locations and institutions represented today. Today's session is being recorded, as you heard when we started. We will share the recording with everyone who registered, and it'll be posted to the ABRF website as soon as it's processed. Just a couple of quick announcements from ABRF. All of you may have received your 2023 membership renewal. Please keep in mind that renewals are completed by the end of the year. You have the opportunity to bundle your annual meeting registration with your membership and save a little bit of money and join us at the meeting in Boston next year. And if you have your phone, you can scan that QR code or just go to the ABRF website to learn more about renewing your membership. Quick one-question survey that we want to learn more about the other societies that you belong to, because that provides us a way to explore collaboration opportunities. The executive board has established a task force to look at collaboration opportunities with other societies. So learning more about which societies you also participate in will help us identify the natural connections with other peer groups. So again, if you have a moment, take a picture of that QR code or follow the link in the slide just to give us feedback on the other societies that you're a part of. And finally, I want to remind everyone that registration is open for the 2023 ABRF annual meeting, May 7 to 10 in Boston. We're really excited about that location. We think it'll be a great opportunity to bring everyone together. Special early registration rates are available now through the end of the year. And once again, you see the link and the QR code to register and learn more about the program as it's coming together. All right, with that, I will stop sharing now and turn it over to our colleagues at Curio Bioscience. Bertrand, it's all yours. Thank you.
Thank you, Ken, for the introduction. I'm gonna share my screen. Okay, hopefully everybody can see that. So thank you for the introduction. My name is Bertrand Young. I'm the Director of Business Development for Curio. So thank you for joining today. Just to give you guys some background on who I am, after grad school, I went to BioLegend to work in business development, and I was responsible for in-licensing Slide-seq into the company and then building that product line and all the panels that went along with that and all the products. And then earlier last year, I joined a company called Spatial that was commercializing seekfish. And so I'm very familiar with the fish-based technologies. And then earlier this year in April, I joined Curio. So very happy to be here. We have a great product to go over today that we're making available to people. And what it is a whole transcriptome, high resolution spatial sequencing platform. So a little bit about the company. We're a life sciences tool provider, pretty young. We were founded in February of 2021 and headquartered in Palo Alto. It's a really great group of seasoned industry and academic leaders that have had a lot of experience in single-cell genomics and molecular counting. And this group of people have come together and try to really create a robust spatial genomics platform for the public based on technology out of the Broad Institute called Slide-seq. Our mission in general is to provide really robust and great spatial genomics solutions that are at high resolution. And we really want to lower the barrier of entry for people to access this new technology. And I'm sure many of you are very familiar with why we do spatial, but just for those of you who aren't, in the past with the advent of unbiased RNA sequencing, people were able to look at bulk samples, taking biopsies and crushing up the tissue and extracting RNA. But they were really missing a critical piece of information, which was what is that sample entail? Like what are the populations of cells? There's really wasn't a way to tell because it's just an average expression profile for that population. And so, with the invention and implementation of single cell several years ago, this is now possible. But again, it was still missing a key piece of information, which is where were the locations of all those cells in those populations. And so in the last few years, it's been an explosion in new spatial technologies, as I'm sure many of you are aware, where now we can measure molecular entities in a spatial context. And so even though there's been a lot of these new technologies coming online, there still have been quite a few challenges that have remained. And I've just listed a few of the major ones here, but one of the main ones is there just seems to be some trade-off between resolution and the number of targets and there doesn't really seem to be a technology yet that can do both at the same time reliably and robustly. A lot of times also the workflows can be cumbersome, so that really prevents the ease of implementation into many institutions and core labs where it's not really amenable to high throughput or atlasing projects where you really need to pump out data very quickly. And a lot of times there is the need for assay optimization for different tissues for various reasons. And along with that, there can be very high assay to assay variability. One of the main things that is preventing a lot of broad adoption of these technologies is also the existence of very expensive capital equipment that may need to be purchased up front in order to use a lot of these technologies. And so what we're trying to do is address all of these challenges and to make a product and to release a product that is easy for people to use and fits right into current pipelines, current sequencing and single cell pipelines. And so we are introducing the Curio Seeker kit, which is a whole transcriptome high resolution spatial analysis tool. And these are some of the features or important features of the product. The resolution is 10 micron. And this is enabled by 10 micron beads, which are randomly placed on a tile or an area that we call a tile. And this tile is 3 by 3 millimeters. The plexity is whole transcriptome. We do have oligos on the surface of the beads that have poly T on the ends that will capture in an unbiased manner all the mRNA with poly A and poly A tails. We don't require any specialized hardware, so you don't need to purchase any instrumentation up front. This fits right into your single cell analysis pipeline. The only thing, a single analysis pipeline. So all you'll need really is a PCR machine, tape station, and sequencer. There's no peripherals, no accessories that you'll need in addition to that. Sample types, we currently support fresh frozen tissues. A lot of people ask us if we support FFPE, and we are working on that, but there isn't a timeline right now for when that will be available. Throughput is really user-defined and highly depends on how comfortable you are with currently existing single-cell pipelines or workflows and or genomics workflows. Generally, if you're very familiar with them, you can do quite a lot of samples at once with one person. We have some customers that can do up to 20 samples in one go. If you're less comfortable with the assay, even if you have no experience, the assay is easy enough that you should be able to at least run 1 or 2 at a time until you get more familiar with it. In my case, personally, the first time I ran this assay, obviously I've had a lot of experience in single cell when I was at BioLegend, but I was able to run 6 at once pretty comfortably. So it wasn't too much of an issue and the results were great. The workflow itself is 8 hours with about 2.5 hours of hands-on time. The rest of the time is mostly just incubations on heat blocks or waiting for the PCR machine to run. As for software, we do have a dedicated primary analysis pipeline that's available for installation locally on your HPC or whatever you're using for analysis. And what this software does is basically convert the FASTQ to matrix files. So we'll take your FASTQ along with a text file for that particular tile with all the spatial barcode locations, and it'll spit out an HTML report with some QC information and some basic UMAP visualizations and top 10 differentially expressed genes. But as well as a Seurat file output if you decide to use Seurat for your secondary analysis, but also a generic matrix file if you have a secondary pipeline that you want to use on your own. So this is the actual product. Just on the left, you see the reagents and the tiles in the pouch. So we have a really robust platform. We really want to make it easy for the customer to use. And so we have a rigorous manufacturing pipeline, extensive product testing and QC. If you remember on my last slide, I did say that all the 10 micron beads are randomly placed on each tile. So what that means is that during our manufacturing process, we actually have to index or sequence all those spatial barcodes up front so that the customer doesn't have to, right? So that's very important because we don't want the customer to waste time trying to do that because it's very labor intensive for everybody else to do. But we have a process internally that can do that pretty easily. And so in the kit that we send you, you'll get the spatially indexed tiles and the reagents, but we'll also email you or send you the text files with the actual bead barcodes and spatial locations. And you'll just use those text files as input into the primary analysis pipeline that we have. So on the right, you'll see the actual product. In the middle, there is a white patch, which contains all the 10 micron beads in a 3x3 area, 3x3 millimeter area. And there are about 80,000 beads in that small patch And those beads are mounted on a glass backing, and that piece of glass is actually mounted on a blue piece of tape that isn't very sticky. It's kind of post-it level stickiness, but it's just meant to prevent the tile from moving around too much while you're mounting the tissue. And that blue piece of tape is mounted in this white plastic cartridge that's about the size of your normal slide. And what you'll do is you put that into the cryostat ahead of time to come to the same temperature as your tissue block. And I'm just going to show you an example of how to mount your tissue. I'm going to skip to the middle. But basically, there's a 10 micron section for reference. This is a mouse brain, whole mouse brain, and it is larger than the actual tile area, which is not a big deal. All you'll need to do is position it over the tile in whichever area you want to look at. And it's easy because it's at the same temperature as your tissue block, and your tile is at the same temperature, so it won't melt preemptively. And so you just move it over the tile to wherever you want it within the tissue. And once it's in position, you just pick it up and melt it with your finger. Right. So it's pretty easy. What you'll do after this is if you are sectioning multiple tissues, you can put this back in the shipper that came with the tile and keep it in the cryostat sealed so it doesn't evaporate too much while you're sectioning other tissues. And you can also keep it up to 2 or 3 days in the -80 if you happen to section this on Friday, you want to process it on Monday, or if you're sectioning a bunch of tissues up front before you process them all at the same time. Oops. And what you'll do after mounting the tissue on the tile is you'll take a pair of tweezers or forceps, and you'll peel that glass tile off of the blue tape and put it directly into an Eppendorf tube with hybridization buffer. And it'll sit there for about 15 minutes to bind, and then you'll put it in an RT mix in the tube as well. And then you'll add some tissue digestion buffer after that to completely digest the tissue and get into solution. And that may involve some pipetting up and down to just get the tissue completely dissolved. But what that does is allows you to have access to the beads. And so you'll use your pipette to squirt the buffer against the beads on the tile, and they'll pretty easily come off at that point. And then what you'll do is you'll take that glass piece out of the tube, and the rest of the protocol is just centrifuge, spin downs of the beads, washing, and it'll be just in Eppendorf tubes or PCR tubes for the rest of the protocol. So very straightforward, not a lot of manipulation on the customer's part. And what you'll expect is this type of data. So here I'm just showing the total UMI counts for various beads on the tile. And the number of UMIs that you'll get per bead will vary quite a bit depending on the tissue and where you are within the tissue. So at least in this case, there are certain areas without cell bodies and certain areas that do have a lot of cell bodies. And then you'll have tissues that are very transcriptionally active. And so you'll see a lot more capture per bead. But in this particular case of the hippocampus and the mouse, you're seeing 6,000 to 7,000 UMIs per bead, and this equates to about 1,000 to 2,000 genes per bead. And across the whole tile, if it's completely loaded, in this case, you're getting about 18,000 to 20,000 genes in total. If you take it through secondary analysis pipelines, you can look at genes individually pretty easily. You get good signal to noise, and you can very clearly see structures within the tissue. And one thing, I'm going to go through these pretty quickly because the main point here is that I just want to let you guys know that there's no optimization that you'll need for any particular tissue. It's the same exact protocol for whatever tissue you're doing in human, mouse. We've had some people do planaria, zebrafish, any species. We've had people do chicken. And so you don't have to worry too much about optimizing this for whatever projects you're working on. And this is just an example of mouse liver, looking at various genes, and mouse kidney, good capture, and again, looking at various individual genes within the kidney. So like I mentioned earlier, this commercial product is based off the Slideseek V2 technology from the Broad Institute. We have internalized this and improved upon it quite a bit, just to make it more robust and easier for people to use. Most people have reacted very positively to the kits that we have sent out to people already. The technology that it's based on, Slide-seq, has also been validated in literature quite a lot. And as recently as October this year, there was a paper that came out from Cornell in Nature that actually used our Curio Seeker kits for their analysis. If you'd like to get in touch, you can go to our website, which is curiobioscience.com. And at the very bottom, if you scroll to the bottom, there's a form that you can fill out. And if you fill it out, we'll send you a protocol immediately, and you can get in contact with us to get access to these kits. And with that, I'll end here, and I'll take any questions at the end. Thank you.
All right. Thank you, Bertrand. I'll pull up my slides here. All right, hopefully you can see my slides. So hi, everyone. My name is Kate Hall, and I'm working the Sequencing and Discovery Genomics Core at the Stowers Institute for Medical Research. And today I'll be telling you how we offer Curio Seeker spatial transcriptomics method as a core service. Stowers Institute is in Kansas City, Missouri, and we're a basic biomedical research facility. We work with model and novel model organisms such as planaria, which are little flatworms, cavefish, nematostella, lizards. We don't do any human work, and mouse is really the most normal organism we work with. So with that in mind, the considerations that we had for implementing a spatial transcriptomics workflow at Stowers really needed to be a solution that was compatible for any organism, suitable for discovery work, This would mean an unbiased approach that captures mRNA, or probe-based with technologies that might allow custom probe sets for our weird little critters, but those tend to be expensive. And we need it as close to single cell resolution as possible. Our tissues tend to be small, so a smaller capture footprint is okay. Fresh frozen tissue works for us, and like all cores, we need it to be affordable. You may have seen me or others from Stowers present in the past on how we were using the published method Slide-seq from Fei Chen's lab at the Broad Institute for our spatial projects. We liked this approach because it is unbiased, using oligo DT capture. It uses barcoded beads with 10 micron resolution, can start with fresh frozen tissue, and being from a publication, this method was extremely affordable. So it was checking all the boxes that we needed and was a great place for us to start at Stowers. And also Fei Chen's group has been absolutely amazing to work with and so very generous. But of course, coming from a publication, you can appreciate that all the reagents had to be ordered from various vendors, and there was no company to talk to for tech support. So we were really excited when we heard that the Slide-seq method was essentially going to be commercialized by Curio Bioscience. This method was clearly checking all the boxes that I had already mentioned, being unbiased and micron resolution and relatively affordable. Being a commercial product from a company, it's of course going to be more expensive than a homebrew method such as Slideseek, but the price tag is not too high considering that probes and custom probe design is not required. And importantly, like Bertrand mentioned, there's no capital equipment needed, which is really important for core facilities to know. And on top of these checks, we're also gaining that the kit, it comes in kit form. So no more ordering individual reagents, and there's tech support. There are people to talk to that can really assist if you need any help. And there's also the layer of flexibility baked into the workflow, which Bertrand touched on and I'll mention later. And it's also really fast with the whole wet lab workflow from tissue to sequencing ready library being easily done within 2 working days. So one of our labs came to us wanting to do a Slide-seq experiment looking at the immunosuppressive microenvironment of colon cancer in mice. And this was right when we received a kit to test the Curio workflow. So we thought this would be a great opportunity to do both methods side by side to help evaluate Curio Seeker product with our established Slide-seq workflow. And for this talk, I'll just focus on the Curio workflow part. But the Slide-seq part was performed according to the published method, and you'll see some comparison of the data later. And I will say the information from this project is currently unpublished, so we have agreed with the lab that we won't go into too many specific details. It might be a little general. So this lab is studying intestinal cancers, and they were interested in the spatial context of tumor tissue because of the complicated tumor microenvironment, including both innate and adaptive immune cells, as shown in the image. This heterogeneity in the tumor is one of the main reasons for drug resistance. So seeing the spatial pattern for how these cells are arranged and respond to treatments can be really important. So the lab provided 4 tissue samples from B6 mice injected with MC38. There were 2 colon tumor samples, 1 treated with a myeloid antibody and 1 control. And there were 2 spleen samples, again, 1 treated with a myeloid antibody and 1 control. And this is the basic workflow of how we approach the Curio Seeker workflow at Stowers, starting with the planning process and tissue optimization on the left side. And once the researcher has worked with our histology team to optimize the tissue sectioning, we move forward with the real experiment, which is what I'll focus on. And I show this chart just to illustrate how much of a cross-core collaborative effort this workflow is. It really utilizes the expertise of many of our core facilities at Stowers. The first step in our workflow starts with our histology core. They'll embed and freeze the tissue so that it's fresh frozen. Then they'll section and place the 10 micron thick tissue section of interest on the Seeker tile. And these first images are what the Seeker tile looks like Bertrand showed. Now it's important to note that images cannot be taken of the tissue once placed on the tile. So our histology team will actually collect adjacent tissue sections for H&E and DAPI staining to help inform later analysis. This will kind of be going on the side, like a little offshoot workflow that we just choose to do. They'll collect and stain these adjacent sections and give them to our microscopy core for imaging and scaling. And you can see the tumor images on the top and the spleen images on the bottom there, with the area of interest circled in each, and that's where the target to be placed on the Seeker tile is located. So these should be almost identical to what the tissue on the tiles looks like. And this initial step is also where the advantage of flexibility comes in that I mentioned earlier and Bertrand mentioned as well. You can absolutely adhere the tissue section to the tile and then freeze it at -80 for a couple days if you don't plan to continue processing immediately. So that's really helpful. I'm now going back to the sections that go on the Seeker tiles. Our histology core will then hand the project off to us in the sequencing group, and we'll do the mRNA hybridization, cDNA synthesis, library construction, and Illumina sequencing. And this whole process takes less than 2 working days. So it's a really quick workflow, which we really appreciate. We create an Illumina library with a unique I7 and I5 index. Read 1 will be the spatial bead barcode and UMI information. We aim for at least 200 million reads per tile as suggested by Curio and run these on any Illumina sequencer based on the number of reads needed. Once the sequencing data has been generated, our computational biology core then takes over to do primary and secondary analysis. For primary analysis, they use the Seeker pipeline from Curio, and then they can use packages such as Seurat from Rahul Satij's lab for secondary analysis. For the initial pre-processing analysis steps, the Seeker pipeline facilitates these key steps. I won't go into too much detail, but I can give a little overview if we have any computational biologists here. So first, you'll tag the read2 sequence using the spatial barcode and UMI sequence, filter the reads based on read quality score, then trim the reads, like you can trim out the start sequence and the poly A tails if you'd like, and then convert the BAM files to FASTQ for alignment. Once the pre-processing is complete, you can then align the RNA sequences, assign the mapped reads back to the spatial location, collapse the read counts down to unique UMI counts, and finally generate the report giving an overview of QC information, statistics, clustering, and spatial patterns. And as I mentioned, we had done this particular project comparing Seeker with our existing Slide-seq method, and here we show some overall stats of the genes detected and beads detected from the 2 methods. And Curio is reported in the pink color and Slide-seq is in the teal color. And as you can see in the table, Curio detected about 23,000 genes for each sample, while Slide-seq had around 20,000. And the beads detected was almost twice as high for Curio in all samples. And our analysts made the plots on the bottom, which are on a log scale showing the UMIs per bead and genes per bead for the 2 technologies. And we can see that Curio is higher for all samples in this case. Now here's the good stuff, where we can see the spatial pattern of the tissue. So this is a spleen control sample, and you can clearly see a beautiful spatial pattern in the image on the left, which is the Curio result versus Slide-seq on the right. And as highlighted in the table in the box in the middle, the median number of genes per bead is significantly higher for Curio. So this was really exciting to see. The treated spleen sample, we also see the spatial pattern looking great and distinct. And again, the median number of genes per bead being much higher. Same story for the tumor control sample, looking so much better with Curio. The spatial pattern is much more defined. And then lastly, the tumor treated sample. Again, same story here. We were really excited when we saw how improved these results were when just looking at the whole project on this first pass of analysis when compared to Slide-seq. Overall, Curio seemed to capture more distinct transcripts compared to Slide-seq, and it had a higher percentage of usable reads. Usable reads meaning that they were of high quality, matching a spatial barcode, mapping to the genome, and aligning to the gene region. For example, here, the colon tumor sample, we have a high percentage of usable reads for Curio at 38% compared to Slide-seq at 23%. For the other 3 samples, we see that same pattern, which is very encouraging. So now we can briefly take a look at how the spatial data can be used to address the question of our lab's project. One thing that can be done for all spatial projects is you can generate a single cell RNA-seq data set on the same type of sample to integrate and help provide more information of the cell types and transcripts being detected. So for instance, here for the tumor sample, on the left, the spatial data was able to identify 9 different clusters, but the lab did have a single cell RNA-seq data set as well in the middle that had clustered into 18 different cell types. And now on the right, when they combined the 2 data sets, they were able to just combine everything and give the spatial view an even more defined look by introducing all the different cell types. One example of something really cool that they saw spatially is here with the colon tumor samples. They were able to use the single cell RNA-seq data to assist in identifying what were tumor cells versus myeloid cells and where they were located in the tissue. And when they merged this data on the far right there, they could see that the myeloid cells remain in the peripheral region of the control sample, but the sample that underwent the antibody treatment allowed for the infiltration of myeloid cells into the tumor region. So that was just a really brief example of the power of this data, because in the case for the colon tumor sample, they were able to confirm that the antibody treatment did change the tumor microenvironment from immune excluded to inflamed, allowing other cell types to infiltrate, which is on par with what has been shown in other publications. And again, that was just one example from the 4 samples that they were looking at. So in summary, I hope I've demonstrated how we at Stowers have implemented Curio Seeker spatial transcriptomics method using collaborative efforts of a few different core facilities. The 10 micron resolution, the mRNA capture, and the fact that no capital equipment is needed all make this extremely easy to implement and also affordable. Our side-by-side comparison with Slide-seq has shown that Curio seems to capture more distinct RNA molecules. Curio detected higher UMIs and genes per bead, and also Curio has more clear spatial patterns. And we were really impressed with this first set of data, and we have moved forward already with other spatial projects using the Seeker kit. And so far we've done projects on mouse fetal liver tissue, just waiting for those results, the liver fluke, which is a kind of parasite. And tomorrow our group member Caitlin is going to be starting a project on chick embryo tissue. So we're really excited to tackle more projects and look forward to seeing some more great data. And with that, I'd like to thank all the folks that made this Curio project possible at the Stowers Institute, specifically Cece of the Lee Lab here, our histology folks, especially Seth, who has done the tissue handling for all our spatial transcriptomics projects. And you may have seen him give a talk on this at ABRF in Palm Springs earlier this year. Thanks to the sequencing team, of course. My cross-copy group, the computational biology core, and our mouse facility members. And a very big thank you to Marie and Bertrand of Curio and that whole team, the whole company. They have been extremely helpful and very wonderful to work with. And of course, need to thank Fei Chen and his lab members who assisted with Slide-seq. And thank you to the ABRF for the opportunity to present to you today. With that, I will stop sharing. And I believe we can take any questions.
Thank you both for presenting. I really appreciate it. We do have a few questions in the chat. One question was to you, Kate. It says, do you also use the 10x Visium platform for spatial transcriptomics? And that was from Sandra Splinter Bondurant.
Yes, we have used the 10x Genomics Visium platform before in the past, not often. The resolution is a little lower currently. I think the spots are 55 micron and there's a lot more space in between them. It's definitely applicable to a lot of other places and tissue types, but for the work that we do at Stowers, really needing to be discovery type of work, we really need the highest resolution possible. So we've been sticking with these 10 micron type of technologies. So we can offer Visium. The project type that would benefit from it just doesn't come along that often here at Stowers.
Great, thank you. And then this one could be for each of you. How many UMIs and genes can be detected per bead? So Kate, I think you had a slide on that.
I'll have to pull it back up. It's not on the top of my head, but if Bertrand, if you have a
Yeah.
answer.
Yeah, it really depends. I mean, there isn't really a solid like hard number for that because it really, as I said, depends on the tissue type that you're doing and where you are within the tissue. So in the figures that I showed, in the hot areas, you're getting, at least in the brain, you're getting 6,000 or 7,000 UMIs per bead, or 1,000 or 2,000 genes per bead. And in the liver and kidney, which are more uniform, relatively speaking, overall, you're still getting 6,000 or 7,000 UMIs per bead. And it really depends. Like there's some samples, like in tumor samples, that are more transcriptionally active, that you'll see much higher numbers. The capacity of the beads is much higher even than that. So we're pretty comfortable with knowing that whatever's there in terms of mRNA will be captured by the beads.
And in the example that I showed, looks like we were getting a mean UMIs per bead was around 500, but definitely went up into the thousands on the high end. And again, that was for these particular tissue types though.
Right.
Great, thank you. Bertrand, there's a question for you. It says, can you add a stained image of an adjacent section, which I think this may have been answered, so that they can use the overlay of the UMI info and the structure in the tissue to put the gene expression in spatial context, as is done with Visium. And I think, Kate, you showed that as your serial sections being done by the histology core to be able to overlay those. And if you have anything to add to that, please jump in.
Yeah, so basically, as Kate mentioned, you cannot image the tissue, the same tissue section, because it's mounted on the beads. The beads are opaque, so it's very difficult to image through it. But you can use adjacent sections for sure. A lot of people, or people who need it, will do an H&E stain of an adjacent section. But for the most part, I'd say most people haven't needed to do that, because as long as you know genes responsible or very highly expressed in certain structures within your tissue, you should be able to clearly see what those structures are just with the gene data. But obviously there will be tissues that are less well-defined structurally. So in those cases, you can take an adjacent section.
And I think it'll depend on your experience too, and what type of facilities or equipment you have access to. We had an established kind of workflow already to go ahead and collect these adjacent sections, stain them, and give them to our microscopy core pretty easily. So if you're able to do that yourself or within your own group, or you have the resources readily available, it's not difficult, and it can help.
Great, thank you. We have a couple questions about the cost, and then there's sort of a multiple piece question. So I think we can talk about the cost first. And Bertrand, if you want to talk about the kit costs.
Okay, yeah, so yeah, thank you. So the kit itself includes 8 tiles. They are all individually mounted on their own tile or slide, basically. And it includes all the reagents that you'll need for library prep. So that kit is $10,000 for those 8 tiles and reagents. So it's about $12.50 per sample or tile. Just a note, you'll need the Illumina Nextera XT kit on top of that, just for the tagmentation at the end of the assay. And then separate from that, there is a dual indexing kit that we sell for $400 that will cover you for 40 samples. And you'll need that because it's specific to the PCR handle on the bead portion of on the bead.
And then Kate, if you could comment, they say, someone says, could you comment on the associated costs, consumables, labor, staff time, etc. Also, if there was not an integration between different core facilities, is the workflow compatible with the researchers handing off samples for library prep and sequencing, and at what step?
Yeah, so for the cost, like Bertrand kind of touched on, the consumables are very minimal. You're working in single tubes for a few samples. The Nextera XT kit is needed. The labor, it's really short. So I think we do add on maybe a couple $100 just to the price per sample, but I can get back to you with a more defined answer about that if needed, about what we do. Our institution's a little different, being a non-for-profit and the way we're set up financially. And if there's not an integration between different core facilities, I think one great thing that we both touched on is just the ability to section the tissue, place it on the tile, and then you can keep it frozen at -80. So you could transport it to different facilities to hand it off for library prep and sequencing right from the very beginning. Otherwise, if they're part of the kit, you can do the cDNA synthesis step, and then there's a safe stopping point where you can keep the sample stored if you wanted to hand it off then to a different facility to do the library prep and sequencing, but you'd have to split the kit or the barcodes, I guess. But the workflow is not too cumbersome, as Bertrand mentioned. So if you can, do these steps yourself at the bench, you could get it all the way through library prep and then give it to your sequencing core, I would think.
So that's all the questions that are in the chat. Ken, do you usually open this up to people to ask live questions?
No, I think we do rely on the folks that post questions through chat or Q&A, primarily
Okay.
through chat. And if we've answered everyone's questions for today, then I think we might be in good shape.
I do actually see a couple questions in the Q&A.
Oh, yeah, I thought those were addressed. So,
yeah. So Kate, I think there's one for you. It just says, how many samples do you recommend batching at a time for a core facility?
And I think you touched on this too. It's kind of what you become comfortable with once you do it. Our core facility, we've tended to try to aim for around 4 or less if the project is less. We haven't had a project come to us that has required more than 4 at a time, I'll say that. But part of the steps are centrifuging your samples to pellet the beads and then pulling supernatant off. And so I feel like the more samples you handle, the more chance the pellet is kind of coming apart maybe a little bit. I don't know that that's been tested and proven, but that's my fear at the bench, is like I always want everything to stay really compact and tight and pelleted, and I feel like if I have to handle too many samples at a time, I might risk that a little bit. So our particular core, we're pretty comfortable with 4. I think with this workflow being as streamlined as it is, we would probably be comfortable going a little higher, but you do just need to kind of work one tube at a time. So you just need to keep that in mind.
Yeah, thank you.
Great, thank you. And then there's one more in the Q&A for you, Bertrand. Can you describe how the Slide-seq or Curio tech works? I'm curious about the transfer molecular biology. Also, how do the beads hold so many transcripts?
Right. So I can't go into too much detail about proprietary stuff, but generally, there are oligos on the bead with PCR handle, bead barcode, UMI, and then a poly T, generally speaking. And so the poly T will capture the RNA via the poly A tail, so that in that way it's unbiased. And like I said, there isn't really any optimization that's needed. And one of the reasons why is we don't fix the tissue beforehand. So you don't need to figure out any permeabilization conditions as you would for other technologies that do have to fix the tissue ahead of time, as if, for example, if you have to do H&E ahead of time. As for transfer molecular biology, I think what you're referring to is how the RNA hybridizes to the oligos. It's only 10 micron sections, so just by diffusion, they should travel pretty quickly to the beads. There is very limited, if not any, diffusion outside, very, very far from the beads. We have some instances where people will partially mount tissue, as you've seen here, where you can clearly see the border of the actual tissue. So there isn't really much evidence for much diffusion. As for the number of transcripts that got captured by the bead, we manufacture these beads and synthesize the DNA on the beads ourselves. So that really depends on our manufacturing capabilities. And we've gotten it to a point where we're comfortable with how many transcripts that it's capturing, which is pretty high. And so I think most people should be pretty satisfied with that. I hope that answered the question in general. Thanks.
Thank you. We got one more in the chat that says, what is the roadmap for the product? Are you working on a larger tile area? And you already mentioned the FFPE samples are in the works. So if you have any updates on that.
Yeah, so yeah, we are working on FFPE. As you know, it's generally a difficult problem to address. We do prefer in general to keep this unbiased because we really believe in the discovery potential of what we have, and so we want to really push for that. But we don't really know when we'll be able to get that out to people in the future. So timeline's kind of open on that one. As for larger tiles, I personally go around to labs, or I've been to the HCA meeting, the Human Cell Atlas meeting, and others, and all of them who are working on humans, but even for mouse tissue have requested larger tiles. So we have been seriously looking at that and what that would entail for us manufacturing wise. So we're really seriously looking at it, and if we do it, no guarantee, if we do it, it probably would be in the range of like a 10 by 10 millimeter or something like that.
Great, thank you. I think that's all the questions we have. Wait, let me look. Maybe one more. Someone wrote 10 by 10 would be perfect.
Good to know. Thank you.
Thank you for that input.
And I think we have one more for Kate in the Q&A.
Oh, yep. Oh, that's right. Kate, do you find that having an integrated computational biology team is critical, or can researchers handle these data sets on their own?
Sure, and I'll let Bertrand can speak to this too. I'm in the wet lab part. I'm in the sequencing team, so I don't handle the data sets to be able to say. I think it's definitely a great advantage to have a computational team because they get used to the workflow. They flesh out all the details, and you can always feed the data, any data from any project of your institution, directly into their pipeline, and they can do everything very consistently and know what they're looking at. I do know pipelines and work and software packages are these days being written to be really user-friendly so that a researcher can do everything themselves. I just don't have the experience with that, but if you do have a comp bio core that can do everything consistently and really get a feel for the data, I definitely see the benefit there.
Yeah, I totally agree. I think it's definitely an advantage to have somebody who's very familiar with computational biology. I would say since single cell and even spatial has been out for the last 4 or 5 years, there should be quite a number of people at various institutions who are pretty experienced in this field and should be able to help whoever needs help. That said, we internally, we've developed this primary analysis pipeline because we have to give it to you guys to make sure that you're able to analyze the data. But as far as secondary analysis, that's really, that's a fast moving field. And, we don't want to develop anything internally that we require people to use for secondary analysis. We are working on a very simple secondary analysis tool that will be released online, later next year. That will allow people just to very in the basic sense, look at individual genes, see where they're distributed, that kind of thing, but not to the level of what people will need for seriously and deeply interrogating their data for a paper, for example. So I think for that type of thing, you'll definitely need to get in touch with a bioinformatics core or people that who deal with this type of data in general.
We have one more question. It said, is there a cost for the analysis software?
No, at this time, there's no cost, especially if you just get it installed locally on your own HPC or on the cloud. We will be eventually putting this primary analysis software online on the web so that you can just upload your FASTQ with a text file, and then it'll spit back the HTML and matrix files so that you can put those files into whatever secondary tool you need. We haven't really decided how that's going to be implemented yet, but for now, it's free in terms of getting it installed locally on your own server.
Last call for questions. I think we're set.
Yes, thank you all for participating, and Bertrand and Kate and Jane, thank you. And Anoja, once again, thank you for organizing this terrific series. We appreciate it. We hope everyone has a great day. As I mentioned at the top of the call, this program is being recorded and everyone will receive the link shortly, either later today or tomorrow. So once again, thank you all for participating, and we hope you have a great day. Bye now.
Thank you.